Okay, thanks everyone and welcome. I'd like to begin by acknowledging the traditional owners of the land on which we meet today, and I would also like to pay my respects to elders past, present and emerging. Well, good afternoon everyone and welcome to the August FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of each month. And last month in July, we heard from Associate Professor Fazil Shabanpour, who is a group leader at the Florey in Melbourne and also CSO of Centron Bio. Fazil presented on his pipeline of antisense oligonucleotide therapies and peptide modifications to allow better access to the blood-brain barrier. We didn't record this seminar, so it's not available for viewing on our website. However, if you are keen to learn more about this, please do reach out to us and we can help direct you to more information. Following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and can read out questions on your behalf. Please note that the chat function is only enabled if you're in the app-based version of Teams and unfortunately doesn't work if you're watching from the web-based version. Please ensure you introduce yourself and where you're from before asking your questions so we can get to know each other. We are going to be recording today's presentation, so if you know of anyone who's not able to join live, they will be able to watch it back on our website at a later time. So now to our presentation for today. Dr. Andrew Phipps is a lecturer and research fellow at the Wicking Dementia Research and Education Centre at the University of Tasmania. He completed his PhD in 2019 at the University of Tasmania, where he developed methods to isolate neurons and study changes in chromatin modifications and DNA methylation over the course of ageing and disease. Since 2021, he has had a leading role in Professor Anna King's lab, conducting a preclinical trial of a drug aimed at preventing axon degeneration in models of MND and developing omics-based methods for understanding neuronal degeneration. On top of this very important work, Andrew is also a professional landscape photographer, and I'm sure he would love to show you some of his work. Thank you so much for taking the time to share with us your work today, Andrew. Over to you.
Thank you. Thank you so much for having me here today. It's absolutely wonderful to be able to get the chance to share some of this work that myself and our amazing team here at Wicking have been doing. And I think I'd love to start off by acknowledging the traditional owners and the custodians of the lands on which I'm presenting from, the Muinina and the Palawa peoples of Tasmania, and also pay my respects to their elders past and present, and the wonderful Palawa community that is here in Tasmania today. I'd also like to, first off, acknowledge the amazing team that I get to work with here too. We have a wonderful team of researchers for motor neuron disease here in Tasmania, and some amazing collaborators both here around Australia and internationally. So first off, I want to try and get everybody onto this page of that axons are really important to study. And they are such a fascinating structure of our nervous system. And they're incredibly complex. And so on these first couple of slides, I really want to go through and get everybody onto that same level of appreciation of them. And so they form connections between individual neurons, between different brain regions, connections to our muscles, our gastroenteric system, and so many other functions that they allow us to do. And on the right here, I've taken an image and a GIF here from the Allen Brain Institute that's showing the individual connections here. Each of these different colors is one neuron and showing the path of their connections from their neurites, their axons and their dendrites, and how they connect between different brain regions in this mouse brain projection. So that really shows some of the complexity that you can see here of these axonal and neuron structures. And so why, again, do we want to study them? This is because it's incredibly important for communication. And there are multiple different ways that axons allow us to be able to communicate between our brain and our muscles, which is really important for motor neuron disease. And so I really love, there's Glennon's in 2014 had a really wonderful study that showed and just sort of gave light to some of the different methods of communication and putting some of that in context. And so if we were to rely on diffusion, it would take about 5,000 years to transport molecules along a 1 meter long axon. And then when we take some of the other methods of transport that axons allow us to do through microtubule transport or protein synthesis, you can see how diverse these methods can be for communication, and that action potentials can travel down an axon at 1/100th of a second, whereas something that needs to be actively transported along the microtubules can take up to 12 days. And so axons need to have this degree of independence and degree of knowing what signals, what molecules need to be present within those axons for their normal function and maintaining their normal function. So then where does this come to with motor neuron disease? And so there have been a range of different studies that have shown that axon degeneration occurs early in motor neuron disease, both through post-mortem studies of human tissue and also through a number of different animal and in vitro-based studies. And there's been emerging evidence that's shown that axon degeneration mechanisms are actually distinct from the cell soma. And so that you can protect the cell soma, and studies have tried using different methods to protect the cell soma, but that doesn't prevent axons from degenerating, nor does it restore axonal function and communication in nerve cells. And so we're starting to really see a light of that there needs to be this protection of both, and a synergistic protection of both the nerve cell soma, as well as understanding what's happening in the axon as it degenerates to try and prevent that separate degeneration from occurring as well. And it's really interesting as well that more emerging studies are showing that protein and RNA dysregulation are also critical to the axon and how that degenerates in of itself as well. So Anna's lab and here at Wiking, we've had a long history of working with models of axon degeneration and trying to understand what's happening in axon degeneration in MND. And we've been using models such as canic acid to understand how axons can fragment and how degeneration occurs. And through some of their research, they've been able to identify, them and others, that axon degeneration also causes a change in the structure of microtubules, or the supporting structures that sort of scaffold the axon. And that by stabilizing and protecting those axonal structures, we can prevent axon degeneration or limit some of that axon degeneration. And so you can see some of these plots here where we've treated with canic acid and then provided either Taxol or other different factors that can sort of protect the axon and stabilize these microtubules. And that can help restore and prevent axon degeneration in some of these models. And again, here on the right, you can also see that after canic acid treatment, you can see a loss of the amount of acetylated tubulin. So in these tubule-like structures, there are different modifications that occur to stabilize that structure that are lost as these axons start to degenerate. So how can we go about stabilizing microtubules? There are a range of different proteins and molecules that alter the stability of microtubules and the scaffolding structure of axons. And there are two key classes of enzymes that I have been really interested in studying to try and protect axons from degenerating. And these are histone deacetylases, which remove acetylation from these microtubule structures and allow these structures to degenerate or change their structure and morphology, as well as histone acetyltransferases, which add acetylation back onto microtubules. And this complex nature of addition of post-translational modifications and removal of post-translational modifications allow the axon to be dynamic. It allows it to change its structure, form new connections, be able to maintain its structure over time as we're developing new connections in the developing brain, as well as changes during the aging brain. And so these molecules need to be plastic, but during disease, there can be a loss of that those molecules that contribute to the degeneration or destabilization of these microtubules. And so HDAC6 has been one key target that we've been really quite interested in, as it's primarily located within the cytoplasm of neurons, and its role in the cytoplasm is to deacetylate microtubules. There have been a number of different studies that's shown that knockout of HDAC6 can prolong survival in SOD1 G93A mice, and there's a study done in 2013 that showed that, as well as inhibition of HDAC6 has been shown to rescue excitotoxicity-associated axon degeneration, and that was from Kelsey Hansen at Wiking back in 2017. There's also been studies that show that prevention or inhibition of HDAC6 and preventing this removal of acetylation from microtubules can restore axon transport and prevent axon degeneration in a range of different other models, including AD, the APP-PS1 mouse model, as well as in models of MS. So we've identified, us and our collaborators, Ludo van den Bosch and their labs over in Belgium, have identified that ACY738 is a really interesting novel candidate to be able to inhibit and prevent HDAC6 from inhibit HDAC6 and prevent that deacetylation of microtubules. And ACY738, and this is the structure that you can see here, is actually really quite an interesting molecule because of its zinc finger binding site, that it allows the molecule to pass the blood-brain barrier, and it's also quite stable. It can be administered in the diet. And we can acetylate alpha-tubulin in both liver culture cells, and so ACY738 is here, showing that we're able to acetylate microtubules in liver cells, as well as pass the blood-brain barrier and acetylate microtubules in cortical brain samples from acute treatment in mice. So this looks like a really promising candidate. And so there have been some studies that have used ACY738. Members from Ludo's lab in 2017 and 2019 were able to show that ACY738 was able to rescue transport deficits in fuzz iPSC-derived neurons. So these were from patient samples. And also rescue a motor phenotype and improve survival in FUS ALS mice. So Rosette in 2019 was able to show, and this is some of the data from that study, that ACY738 extended survival in these mice and also showed significant weight gain in these models. However, this is only one type of ALS in modeling this disease. And so it's really unknown whether selective HDAC inhibition and using ACY738 is neuroprotective in other models. And we really need to start to understand what's happening in some of our more traditional models, such as the SOD1 mouse model, as well as some of our other emerging models. So this led us to our research question. And so we collaborated with Ludo and developed and got ACY738, and we worked with FightMND to be able to try a preclinical drug trial of ACY738 to protect against neurodegeneration and axon degeneration in 3 different mouse models of ALS. And so the different mouse models that we were wanting to look at include the TDP-NLS mouse model, which is quite a rapid model of degeneration. And this really shows us the nuclear localization fragment being knocked out of this model to show the effects of that acute TDP pathology and the mislocalization of TDP43, and whether ACY738 would be able to prevent the degeneration in that really quite rapid degenerating model. We also used the SOD1 G93A mouse model, as this is a widely used model, and it would be really appropriate for us to be able to compare against other studies and understand what's happening from that familial perspective as well. And then finally, we also wanted to look at the TDP43 Q331K model, as this model has quite distinct axon degeneration. It's quite a long time course. It's a slower degenerating model. And we then wanted to investigate some of whether ACY738 can protect in that model as well. And I'm just putting a note here at the bottom. I'm only going to be able to show a small snapshot of the findings from this study, of these 3 separate studies today. We've looked at different neuronal counts, microglia, astrocytes, all sorts of different measures from these models. But I'm just going to give the key take-home messages for today. So the first thing we wanted to check was, in our hands with ACY738, can this acetylate microtubules? And thankfully, in all of our models here, it was able to significantly increase the acetylation of alpha-tubulin in the spinal cord from the G93A, the TDP-NLS model, as well as the Q331K model. And for all of these studies, we've also included riluzole as a comparator, as well as co-treatment with riluzole, as these are 2 very likely scenarios that are going to occur for any potential therapeutic that's making it through clinical trials, and if it's going to be given in the clinic. So we wanted to see if there are any synergistic or detrimental effects from co-treatment with riluzole, as well as compare if ACY738 can improve the outcomes compared to riluzole itself. So the first set of findings, I'm going to direct everybody to the paper that was published last year, where this is the findings from the SOT1 G93A model, and we were able to show that ACY738 was able to protect motor neurons in the lumbar spinal cord of female but not male MSOT1 G93A mice. And I direct anybody who wants to go and learn more about the findings from that study to that paper, just for time today, because I really want to get into some of the other unpublished data that we have as well. So now let's jump ship over into the TDP NLS mice. And we were able to initially look for motor neuron loss. Let's see, in the lumbar spinal cord, does ACY738 prevent lower motor neuron loss in these mice? And so we saw initially, I'm just going to run you through some of these graphs here. So we saw a significant reduction in motor neurons in the ventral horn of the TDP NLS mice compared to wild type controls. And so you can see here in wild type, for all of the plots that I'm going to be showing today, these are going to be blue for our wild type controls, and then yellow are going to be our transgenics in male and female. And then we also saw a significant three-way interaction between sex, ACY738 and riluzole treatment. And so there was a post hoc analysis was able to show that female untreated versus female riluzole treated mice, there was a significant difference where riluzole treatment, comparing untreated versus treated in riluzole in our male mice and untreated versus treated riluzole in our female mice, riluzole was able to significantly restore lower motor neuron count compared to untreated controls here. Whereas ACY738, either treated alone without riluzole or in combination, there was no significant difference compared to that untreated control in terms of the post hoc. However, there was that three-way interaction that we saw. We then wanted to have a look at neurofilament light and these blood-based biomarkers, which is a really good measure for overt degeneration in these models. And it's also a really good clinical outcome for clinical trials. So it's really important for us to understand what's happening to these blood biomarkers. And again, you can see here that there was a significant increase in neurofilament in our male and female transgenic mice compared to our untreated controls. But there was no significant difference in serum NFL levels between our different transgenic treatment groups here, with either treatment with riluzole or ACY738 treated or versus untreated here. So then that was the core results from the TDP NLS mouse, and we didn't see much promise from the ACY738 treatment there. So we decided to have a look through at the TDP43Q331K mouse model. And so I really want to acknowledge Dr. Fari Kabir, who did a phenomenal amount of work on this project, and this formed a lot of her thesis. And she's actually also graduating today. So after the seminar, I'm going to go and celebrate with her. And so she identified that there was a significant reduction in the lower motor neurons in the ventral horn of the Q331K mice compared to controls. So that's really good. The model is working well in our hands, and didn't find any significant effect of lower motor neurons in the ventral horn of the Q331K mice treated with either riluzole or with ACY738. So we didn't see any effect of ACY738 treatment as a result of preventing lower motor neuron degeneration in this model. However, when we started to dive into the axons themselves, we started to look at both the sciatic nerve, the dorsal and ventral rootlets, and the neuromuscular junctions of these models. And that's where we started to see some interesting findings from ACY738. So we were looking at morphological changes. So from toluidine blue and EM-based imaging, we were looking at the morphology. And you can see here, comparing wild type to transgenic in the Q331K mice, there's quite a lot of sort of disformed, malformed axons in this mouse model. And we actually saw a significant effect of treating with ACY738 to restore that morphology, that circularity of these ventral root axons compared to untreated controls in the Q331K model. And so this is just merged findings between the males and females here. And then when we went to have a look at the neuromuscular junctions, we actually also saw that there was a significant reduction in the probability of fully deinnervated in neuromuscular junctions after treatment with ACY738 or riluzole, or in combining that treatment with both of those drugs together. And so here you can see some representative images showing alpha bunkerotoxin at the top here and showing those deformed neuromuscular junctions. And then we've also labeled with synaptophysin and SMI312 to be able to see whether that's restored in those neuromuscular junctions compared to our untreated controls. And when you look down here, so on this on the y-axis here, we've got the predicted probability of having de-innervated neuromuscular junctions. So you can see here for our untreated controls from females and males that that's really quite high, but then treatment with ACY drops that probability down quite low, as well as co-treatment. And then you can see here in the males that co-treatment actually, when we did the post hoc for that, was also able to further restore the probability of having intact neuromuscular junctions. And then for our biomarkers, we again wanted to have a look at neurofilament light in the Q331K mice, and we saw that there was a significant interaction between ACY738, riluzole, and sex on the serum NFL levels in the Q331K mouse model. And when we dove into that data a bit more and did some post hoc analyses, we could see that there was significant post hoc differences between the female untreated and ACY treated mice, the female untreated and riluzole treated mice, as well as the combined treatment of both of them. And so you can see here is our untreated and compared to treated with ACY738 or co-treated with ACY738 and riluzole. So we were able to see that there is this reduction in these blood-based biomarkers of degeneration in the Q331K mouse model. And I also want to acknowledge Dr. Jessica Collins, who did a phenomenal amount of work with all of our biomarker assays for all of these studies. So that's a starting off whirlwind tour of some of the findings from the ACY738 work. And so in summary, we were able to show that the SOD1G93A mouse model, ACY738 treatment was able to restore lower motor neuron count for that, but it wasn't able to restore neurofilament light levels. And we're still working on following up on neuromuscular junctions in that mouse model, as well as the TDP NLS mouse model. And however, in the NLS mouse model, and this is really quite interesting because we saw no effect of neuromuscular of NFL, of the effects of sciatic nerves or the ventral rootlets when we analysed them, as well as in the lower motor neuron count for the NLS mice. But then when we look at the Q331K mouse model, we did see an effect of ACY738 in axons, in the serum NFL levels, as well as at the neuromuscular junctions. And so there's a lot of data to try and dive into here for this. ACY738 may be most beneficial at preventing this axon degeneration in some subtypes of MND, such as the Q331K mutation, given the different outcomes of our different models. It might also be more beneficial earlier on and targeting early levels or early stages of MND compared to, say, later or more severe types of MND degeneration. And we also were wondering if there might be sex-based differences in the therapeutic responses for our MND mouse models, whether that be due to the different rate of degeneration in our mouse models between female and male mice, or whether there are inherent differences in the pathology and the progression of MND in these mouse models as well. And so we're going to continue examining some of our other cell body and axon degeneration combination therapies, and as well as finishing off and looking at the neuromuscular junctions in the NLS mice and the SOD1 mice. And then we're going to start working on trying to understand if there are other therapeutics or other ways that we can examine mechanisms or prevent axon degeneration from occurring in these models. So that leads me to the second part of my research program that I'm working on at the Wicking Centre, which is, are there other pathways or mechanisms that we can target to prevent axon degeneration in MND? And so for this part of the study, I'm looking at modelling and different methods of modelling axon degeneration, as it can be incredibly difficult to isolate and understand axon degeneration in vivo, and in particular with some low input samples. So if we're trying to look at individual rootlets or samples from in vivo studies, you're dealing with the myelination, you're dealing with a wide range of different cell types and contaminating material, and it can be incredibly difficult to isolate particular tracts of axons for that work. And that's another area that I'm really trying to work on at the moment, is if we can be able to, using in vivo technologies or using postmortem human tissue, can we start to isolate some of these axonal tracts to be able to understand the omics-based mechanisms for degeneration in these studies. And we're also, one of the ways that we can get around that is by using induced pluripotent stem cells and microfluidic devices. And so through Anna's work and the work at Wicking, we've been building expertise in microfluidic chambers for axonal isolation. And Anna's been working on this project and trying to use this for a number of different years. And then when I joined Anna's lab in 2021, I really wanted to see if we can use some of those techniques to understand the molecular mechanisms for axon degeneration with an omics-based approach. Can we identify and isolate axonal protein and axonal RNA for us to understand what's happening as these axons degenerate in a model of axonal degeneration, or MND? And I want to take a note here and thank both the funders for MND Australia, as well as the amazing team that helped make this project happen, because it was a phenomenal amount of work to get to where we are with this project. And so the model that we're using is excitotoxicity. And so excitotoxicity causes axon and motor neuron degeneration in MND models. It contributes to white matter tract degeneration in postmortem human MND, and it changes RNA regulation, transcription, and RNA transport in MND. And so there are also studies that have shown that AMPA and kainate receptor agonists can contribute to motor neuron death and axonopathy in both mouse models and postmortem human MND. And in recent years, there's been some really interesting studies that have shown that the axon can have its own unique molecular profile. So it has its own rates of RNA turnover, its own rates of translation, and RNA can be actually quite stable in the axon, where you would think that it would get translated and then degrade over the course of hours or days. There are some transcripts that their studies have shown that can last for days to weeks in the axon. And so there's quite a lot more to understand in how that axon is dynamic and whether there are unique changes in terms of axonal protein synthesis. And you can see here on the right, this is an image showing the effects of excitotoxicity on axon degeneration over the course of 48 hours of treatment. And so this leads to the second study that I was working on. And so what are these molecular mechanisms of excitotoxic axon degeneration? And can we target these to protect against neurodegeneration? So I wanted to define when unique molecular and biochemical signatures are altered during axon degeneration compared to control axons, and to then confirm these candidates, identify what's changing in the early stages of axon degeneration, and try and see if we can prevent them from or if we alter some of those pathways that are changing, does that change whether these axons are going to degenerate, or does it exacerbate whether that degeneration occurs? And then we were going to perform a targeted screen of selected pathways to intervene in cortical neuron axon degeneration. And so this work is still ongoing, but I wanted to share some of the findings with you today. And so in terms of our experimental design, we've taken healthy donor cells from lines from the Tasmanian Healthy Brain Project, which is an ongoing study at the Wicking Centre. I've taken two healthy lines, and converted them into iPSCs and then differentiated them into cortical neurons, where we've seeded those NSCs into the microfluidic devices and then allowed them to mature for 14 days. After that time point, we've then either left them as controls or treated them with kainic acid. And over the course of up to 24 or 48 hours, we've identified different time points where we start to see the morphological changes associated with axon degeneration starting to occur. So where we start to see blebbing, or these beads on a string morphology that you'll quite often see, and what time points do we actually see the fragmentation of axons occurring? At those specific time points, we've then pooled our chambers and performed and I've worked up some new techniques to be able to pool these chambers and isolate and perform simultaneous proteomics and transcriptomics from these same samples, for us to then get a really nice matched data set of what is happening to the proteome and the transcriptome of these samples before performing a candidate selection and then intervening with some rescue experiments. And I'd also like to acknowledge Tony, Jana, Alex Hewitt and Alice Pebe for their support with generating the iPSCs for this project. So initially, the first step in this project was to define and determine when these morphological changes that occur with axons, when does that actually start to occur? And so we can start to see degeneration in the axons from 6 hours after treatment with canic acid. And we've tried doing a dose response curve over a range of different time points and doses for canic acid, and we settled on 500 micromolar, which is actually quite a large amount of canic acid, but it worked really well in this model to give us a distinct time point for when we start to see a loss of the number of intact neurons, we start to see beating, and we've also started to see fragmentation at that time point. And so we chose that time point for us to be able to really map out and get defined time points for when we're starting to see axon degeneration, when we're starting to see blebbing and fragmentation. And then from those morphological time points, we've actually taken a couple of steps back. We've taken time points preceding when we see those morphological degeneration starting to occur in that model. And so this leads us through to some of the results. So first off, I'm going to take you through some of the proteomics from this analysis. And so this is first off the overall, across the whole time course, what are we seeing? What proteins are changing during excitotoxic axon degeneration? And so there is a range of different pathways that you can identify from doing the proteomic screen that included a range of synaptic markers, mitochondrial markers, actin polymerization. And we identified over 3,000 different protein IDs from this combined proteomic transcriptomic screen, with 84 different transcripts significantly altered across the entire time course of degeneration. And it was really nice for us to see with hierarchical clustering that you can see that there's actually quite these unique clusters that are changing during degeneration. Sorry. And you can see here that there are some transcripts that aren't enriched in these protein samples in the control or 3-hour time point. But then when you get to 10 hours, there is quite widespread enrichment of this cluster of proteins here. And then there are some subclusters that then start to lose that signal again by the time you get out to 24 hours. Whereas there are other subclusters that are only really enriched at 24 hours of treatment with canic acid. And then likewise, there are a number of different proteins down the bottom here that you can see that slowly start switching off as we go through that time course of axon degeneration. Then I wanted to look at some of these early time points. For me, the early time points, the early alterations occurring during the axon during degeneration are some of these key time points that we can start to try and intervene to prevent axon degeneration in these models. And then hopefully identify new therapeutics that we can use to help improve the lives of people with MND. And so the earliest changes that we were observing in the proteome included changes to post-translational modifications, which was really, if we jump back to the FightMND funded part of this project, using the mouse models and the HDAC inhibition, was really quite interesting to see that these post-translational modifications are some of the earliest changes that we're seeing as axons start to degenerate. We also saw RNA binding and splicing, as well as acetylation in particular being altered. Some of the notable proteins that you can see here were altered, included ubiquilin, and also one here that I was really quite interested in is YTHDF3, which we seem to see a dramatic loss of the protein quantification there at 3 hours before it restores at 10 hours post-treatment. And some of the pathways that we were able to identify included pathways associated with protein binding and phosphoproteins, and these were the proteins that were reduced in abundance after treatment at 3 hours, as well as acetylation, synaptic vesicle formation, and the cytoskeleton itself. And you can start to see some of these seem quite logical in that as the axon is starting to undergo these changes over time, what are some of these pathways that are changing? Whereas the enriched pathways included RNA splicing and binding and ER, endoplasmic reticulum associated pathways. And you can see here, these plots here are the pathway, gene ontology and pathway analysis, showing the things that are depleted early or enriched early during degeneration. And then we saw these interesting clusters of proteins when we were looking at string database analysis for these clusters associated with protein degradation and RNA splicing. Then when we wanted to have a look at what's happening, I was really interested and surprised when I saw that there are a number of different proteins that are enriched at this 24-hour time point. At that stage, there's quite severe axon fragmentation starting to occur in these axons, and I thought that it was going to be quite a at that time point, there was going to be not much changing in terms of the proteome. And it was really interesting that when we're comparing 10 hours versus 24 hours at these later stages of degeneration, that there were actually still some quite interesting and dynamic changes within the axons. They saw changes to cell adhesion, mitochondrial membrane proteins, antigen processing, protein transport, and endosome alterations. And again, when we look at these different clusters, you can see the proteins associated with RNA modification. There's a number of proteins here associated with the cell cycle, energy production, and sort of basic molecular functions that you'd see, as well as an interesting cluster around plaque formation and trafficking when we started to dive into some of these clusters individually. So we've gone through and we've analyzed the proteome to this point, and we identified quite a wide range of different changes occurring. But then we've been able to, through the techniques that I've developed, be able to match that with the simultaneous extraction of RNA to be able to identify and analyze the transcriptome. And so we were wanting to investigate if the changes that occur to the proteins are driven from the transcriptome. Are there changes occurring as a result of just deformation of these proteins, or are the transcriptional and the protein alterations actually distinct during axon degeneration? However, this took quite a lot of work for us to be able to optimize and get working, because the bioinformatics processing, dealing with quite low input samples, and it took quite a lot of time for us to be able to, the last actually couple of years for me to really start diving in and getting the best out of this data. And so across the whole transcriptome, we also saw about 80 differentially expressed transcript in the axon during degeneration. And if we look at some of this gene ontology analysis here, you can see for these geo-based terms for molecular function and biological processes, you can see a range of plasma membrane and ion transport changes, alterations in ribosomes, and some really distinct alterations that are occurring there. And anything that is red is significantly altered pathways compared to background. And again, same with the biological processes, tau protein kinase activity, ATPase binding, and cyclic activated cation channel activity. So there are definitely distinct alterations occurring in the transcriptome of the axon as a result of this excitotoxicity that would warrant following up further. And then on the right here, you can see what's called an upset plot. And this shows the number of uniquely identified transcripts at each of the different pairwise comparisons for, say, control versus 3 hours. And when you have these dots here, those are transcripts, that are shared between the 3-hour versus 10-hour analysis and the control versus 3-hour, or here between control versus 24 and 10 versus 24 hours. So there are a number of different transcripts that are altered that are unique to, say, the 24-hour control versus 24-hour time point analysis, but there are also a number that are shared as we go through this axon degeneration. And so when we're looking at the early effects of axon degeneration in the transcriptome, we identified 21 transcripts that were significantly altered, with 17 depleted and 4 that were enriched. And these transcripts included a number of ribosomal RNAs, transfer RNA subunits, and then because we've done whole transcriptome analysis with enough deep sequencing, we're able to identify a number of different microRNAs as well. A couple of these candidates, microRNAs have been implicated in some different subtypes of MND and have been associated with differential vulnerability of MND, as well as SLX1B has been cited as a transcriptional dysregulation subtype similar to TDP43 subtypes of MND, but this is also associated with its own range of ALS subtypes. And the papers that are associated with that are there. As we start to progress through axon degeneration and we're comparing the early stages of degeneration prior to blebbing and prior to the morphological changes versus when we start to see blebbing and degeneration, we identified 32 transcripts that were altered. And again, these were notable transcripts that were also associated with transmembranes, GTPases, and we also started to see a number of other microRNAs like MIR197 here. And this plot here is some of the top changes that we identified, associated and ranked by p-value with the log fold change comparing to the previous time point. So you can see quite dramatic changes in the transcriptome and the level of changes there in the transcriptome between these two time points. Finally, when we went to look at late-stage degeneration, we identified actually the most number of transcripts at late-stage degeneration between 10 and 24 hours, with 41 transcripts altered between these two time points. And we started to see a number of different unidentified or unknown loci that they haven't been named as of yet, or they're of unknown function. And so we start to see a number of other microRNAs again. We start to see ribosomal RNAs and continuing to see those similar patterns, but a lot more of these unidentified loci that are really going to be worthwhile following up to try and understand what their role is in the axon and how they're altered during degeneration. So from here, this is where I'm now starting to pick some candidates. I've got our transcriptomics data, I've got our proteomics data, and we've seen that there are actually quite distinct alterations in the proteome and the transcriptome as axons start to degenerate in this model. And so I'm going through and I'm picking out some candidates to confirm using Western blotting, immuno, qPCR, things like that, to be able to understand and confirm that these alterations in the proteome and transcriptome are true to our axons. And then I'm going to start performing some intervention experiments with synthetic RNAs, some siRNAs, and other different methods to be able to modify the transcriptome and the proteome in these samples. And so for a number of these candidates, YTHDF3 was one that was quite interesting, as we saw a 5.8-fold loss at 3 hours. And this is a M6A reading complex. And there's been 3 or 4 studies that have identified YTHDF family to be implicated in either TDP processing or associated with toxicity in models of ALS, as you can see in some of the information that I've provided here. Another candidate that looked quite interesting was the CLPX2, which had almost a 2-fold loss at 10 hours compared to 3 hours in the proteome. And this is a part of the complexin or synaptin family that are associated with synaptic vesicle exocytosis, and they're preferentially located in the action potential and involved in long-term potentiation. And so a number of these different proteins and these transcripts here, I've got a few others here that I'm really quite interested in following up, are distinct and altered during this time course of degeneration, and are some of the top changes that are altered, or otherwise candidates that are involved or sort of at key sites or integrating key pathways when you're looking at, say, string database analysis and things like that. And on the right here, you can see, for example, with YTHDF3, this is actually a key regulator in whether transcripts are sent off and become translated, or whether they go through a M6A mediated decay pathway to decay that RNA. And so there could be some really interesting changes to this transcriptome as a result of the loss of YTHDF3, for example. So in summary, we've identified that the axonal protein abundance is dynamically regulated during excitotoxic degeneration. Some of the earliest changes in the excitotoxic degeneration include alterations to post-translational modifications and RNA splicing. We've seen that there are a number of different protein transport and changes to cell adhesion occur within the late stages of axon degeneration. And there are also quite distinct alterations to the transcriptome with microRNAs, tRNAs, and a number of unknown loci altered during axon degeneration. And so I'm still working with my team at the moment to perform candidate validation and performing additional informatics analysis and comparing with some published literature as well. And we want to then start identifying some of these candidates for intervention. And so at the moment, this is where we're going right now with our studies, but we're also asking the questions, are these similar with known genetic drivers of MND? If we look at different models of MND, or whether they are different in different subsets of vulnerable or resistant cell types and populations. And with that, I'd like to thank you all for listening and open up for any questions.
Thank you so much, Andrew. That was a fantastic talk. I think you really took us on this fantastic journey about your different projects and the progress in each of those. So I'm going to open the floor to some questions. Please, if you have any questions, there should be a raise hand button at the top of your screen. If you click that, I can enable your microphone and camera and allow you to ask your question. So I guess if there's no questions immediately, I'll kick off, Andrew, just with asking. Just curious about your degeneration experiments with the canic acid. Obviously over sort of a 24-hour time course. I'm curious how that compare whether that's sort of artificially fast or consistent with sort of physiological, what you see physiologically, and perhaps is that might be, I guess that ties with, is that could be a reason why you're seeing differences in the transcriptome and proteome.
That's a really great question. And it is indeed one of the caveats of the study of using that degeneration time point and having quite a rapid process of degeneration. Usually in models published previously, they've used up to 200 micromolar, but there's a lot of variability when we're looking at the axon degeneration and applying canic acid to this model, especially with the induced pluripotent stem cell derived neurons. And so to get a nice consistent time course for degeneration, we tried 25 micromolar through to 1,000 micromolar doses, and really we tried to pick as low a dose that's going to be relevant without having that inert variability and that really sort of different variability between, say, a 25 micromolar dose, where it would, in some models, in some lines, you'd see overt degeneration within a reasonable time course for that, whereas others, it wouldn't actually degenerate in that model. And so it's trying to pick a nice balance there between something that's going to be a reproducible model versus something that is not as such.
Mm-hmm. Right. And I guess tied to that, just some early ideas about why you see that differences between the transcriptome and the proteome.
That's a really great question, and it's something that I'm still ongoing in terms of my analysis. I was expecting to see some more, or in my hypothesis, that I would see some more matched proteins and transcripts associated. It might indeed be, when we go back to that slide that I was talking about, how axons communicate, that some of these changes, some of these transcripts might take a number of days for them to be able to arrive down at the axon. Yeah. And so some of that time frame for generation may not lead to what we want to see in terms of the proteome itself. But it could also be that they are distinct changes as well, that some of these microRNAs that may be able to travel down further or faster are altered earlier in this time course of degeneration. But there's still a lot of work to be able to do in that space, absolutely.
Yeah, absolutely. I guess according to like your images that you showed of that degeneration time point, that looked like it by 24 hours they were pretty much completely degenerated. So you can't really stretch that out longer to explore that. Is that correct?
Yeah, well, I think if we dial that dose and we try again with a lighter dose of canic acid and then work with that model some more, we could start to see and sort of unravel some more changes. But we really wanted to get a really nice starting point here to see what is changing during degeneration in the first place, because this hasn't really been looked at before. There have been a couple of studies that have used axon-seq to be able to label and identify axonal transcripts in versus, like, say, SOD1 versus control, but we haven't really started to look across a time course of degeneration before like that.
Yeah. Okay. Thanks. A question from Double. Please go ahead, Double.
Yeah, hi, Andrew. Thanks again for sharing the results. Yes, I'm from FightMND, as most would know on the call. I've got a few questions, but I'll go with one specifically over the next steps on this, in terms of, okay, you've identified some targets, potential targets in the proteome, transcriptome. Is your next step to kind of potentially target these areas individually, or to look for common things that might be happening to, further upstream in the pathway to
great question.
Target there?
Yeah, and so it's actually a mix of both. I want so in my candidate selection, I'm looking for common pathways, and maybe looking through, say, string analysis, or looking at these biological pathways and what might be a common driver of, say, multiple of those pathways that lead to some of these downstream consequences, but also really interested in looking at some of the individual candidates themselves, say the top changes. And so I'm picking out a number. I'll get to pick about 5 to 6 different candidates to perform intervention experiments on. That's what I've got budget for anyway at this stage. And I'm really keen to pick a number of these pathways that are either key common factors or some of the direct changes that are happening as a result. And then at this stage, just looking at applying them individually, and that's what we're going to be starting to do for the next the duration of the remainder of this grant, but then looking at other combination therapies and things like that down the line. But I also really want to start understanding if this is similar in different types of cells as well. Are there different pathways associated with axon degeneration in sensory versus motor neurons, lower motor neurons versus cortical neurons? Do they all share that same pathway of degeneration? Is this a common thing? Or are there distinct differences in each of these individual subpopulations of neurons that are preferentially vulnerable to degenerate?
Did you have another question, Davil?
Yeah, I did. Well, I think you've just answered that question, Andrew, in terms of, I noticed that, you've done some sophisticated work here on modeling upper motor neurons, and I was just going to ask, whether those pathways are equivalent in the lower motor neurons and the axons then connecting those lower neurons to muscle. But another cheeky question though is going through back to your ACY studies, ACY 738 studies, and the 3 animal models that you used. I noticed you mentioned toluidine blue in terms of identifying axons. What did you use as a marker of neurons or motor neurons in those studies?
So for that, we used what's it called, crescent violet, to be able to label the lower motor neurons, and then did manual counts from the lumbar spine and using crescent violet for that.
Yeah, right. Can just a question on the NLS model in terms of it's like my lack of knowledge about it. In terms of, is that the docs treated model?
Yeah, that is the docs treated one.
So, and there's a recovery phase in that animal model too, I gather. Once treatment starts, do you know if in that model, in terms of in your crescent, because you're looking at crescent violet, and looking at motor neuron loss, with the recovery phase, do those kind of motor neurons reappear, or how does it work in that mouse model in the recovery phase?
I'd have to go back to the original study to confirm that. My understanding is that once they're gone, they don't regenerate. There's no regeneration of those lower mid neurons once they're lost, is my understanding. But I'd have to go back to the study to double check. I also just remembered too, we also used, had to go back a year there for that. But we did crescent violet counts, as well as also using chat immunolabeling as well for those immunos. For those image stains as well.
Yeah, thanks, Andrew.
Thanks, Andrew. Thanks, Davil. Any other questions from the floor? All right, can I just quickly ask, Andrew, about the sex differences you observed in that earlier study?
Absolutely.
Some ideas about why you're seeing those and whether that would affect further downstream, further studies that you do.
Yeah, and so that's it has been really interesting to see, because we did see some robust sex differences in, say, for example, the Q331K model, which and in both the response for how they responded to treatment, as well as also timelines for degeneration. And we're not sure whether they might be commonalities, or whether it might be either male versus female mice degenerate at different rates. Or whether that's just an effect of the treatment of the drug itself. And it's still something that we really want to tease out a bit more with our studies. And I think it's something going to be important for ongoing studies to really start to analyse sex differences independently. And I think a lot of the research field is moving that way anyway, but this sort of really drives home that point as well.
Thank you. I think if there's no other questions for me, we might wrap it up there for today. So thank you so much, everybody, for joining us today, and thank you, Andrew, for taking the time to lead us on this journey of unraveling some of the drivers of axonal degeneration. It's really exciting to see how far it's come, and look forward to seeing some of the results of your future studies. So thank you.
Thank you so much for having me on.
Not a problem. If there's anything else you would like to follow up with, Andrew, if anyone has anything that they would like to talk about, you can definitely get in touch directly with him, or reach out to us at FightMND if you would like us to help connect you. Again, we did record the seminar today, and it will be available on the website in a few days' time. So thank you so much for joining us today for the August FightMND Scientific Seminar, and we hope you really enjoyed the presentation today. Our next monthly seminar will be on Tuesday, the 30th of September at 12 p.m. Australian Eastern Standard Time. We'll release the details in a couple of weeks' time, so keep an eye out in your inbox for this. Until then, all the best, and if I don't see you at Pactails, I'll see you next month in this seminar.